Hi everyone. In today's video lecture, we're going to discuss about type 1 hypersensitive reactions. This particular topic has been divided into two videos. In the earlier video, we have discussed about what is the definition of hypersensitive reaction and in how many types of hypersensitive reactions are there. And we found that there are four types of hypersensitive reactions. These are type 1, type 2, type 3, and type 4. In this video lecture, we will discuss about type 1 hypersensitive reactions. A type 1 hypersensitive reaction is mediated by a certain type of antigen, which is called as an allergen. And just like any other kind of antigen, these allergens are also going to induce humoral immune response. For example, when an antigen enters inside the body, these antigens will bind to the antibody which is found on the surface of B cell. B cell differentiates into antibody producing plasma cells. But what happens when allergen enters inside the body? These allergens, just like any other kind of antigen, is going to bind to the antibody, which is found on the surface of a B cell. And B cells this time specifically differentiates into plasma cell, which produces IgE. Once these IgE is produced, they are going to bind to the receptors, specific receptors, which are present on the surface of basophils and mast cells. Now, basophils and mast cells are the two very important cells which are going to respond during allergic reaction. So when IgE bind to the specific receptor present on the surface of mast cells and basophils, the person is said to be sensitized, which means the person has become sensitive towards a particular type of allergen. When the same individual is exposed to the same allergen again in the future, maybe second time, third time, or maybe in the consecutive times, then what is going to happen? These allergens will directly go and bind and cross-link the antibodies IgE, which is found on the surface of basophils and mast cells. These cross-linking of IgE on the surface of these cells will lead to degranulation of the membrane, and degranulation of the membrane will lead to release of various pharmacologically active mediators. Now, one of the very common example of these mediators is histamine. I guess everyone knows about it. Now, what are the main effects of these mediators when they're released into the surrounding medium? Like it is going to lead to vasodilation. Vasodilation means dilation of the blood vessels, and also it is going to lead to smooth muscle contraction. This is particular a pictorial representation of the entire process that will happen. Like for example, what happens when allergen enters inside the body? So this is basically the first exposure. The allergen is going to bind to an APC. APC is called as antigen presenting cell, which gets activated. The main function of APC is to activate your Th cells. So when Th cell gets activated, the main function of Th cell is to activate other immune cell, and it is also going to activate B cells. B cell independently also gets activated when an allergen binds to the antibody found on the surface of a B cell. What happens after that? The B cell gets activated and it is going to differentiate into plasma cells. Now these plasma cells, as I mentioned before, they will not produce any other antibody apart from IgE. So they specifically produce IgE class of antibodies only. And these antibodies will bind to the receptors, specific receptors, okay? So they will bind to the specific receptors which is found on the surface of mast cells and basophils. As I mentioned before, once this particular IgE bind to the receptors, this person becomes sensitized. That means the person becomes sensitive towards this type of allergen. Now, in the future, when the individual is again exposed to the same allergen, all right, so it is called as a re-exposure, maybe second time, third time, and so on. Then what is going to happen? The allergen will directly cross-link the IgE, which is found on the surface of basophils and mast cells. And what is the end result of it? The end result is degranulation. You can see the membranes are bursting. So degranulation of these membranes, and it is going to release all the pharmacologically active mediators into the surrounding medium. As I mentioned before, one of the very common example which everyone knows is your histamine. So histamine, you can now know that it is a pharmacologically active mediators and it is a main culprit of causing allergic reactions inside the body. Now, apart from histamine, what else we have? We have leukotrienes, we have other mediators. We'll come across different kinds of mediators in the coming slides. So this is how degranulation of the cell occurs, and these mediators, when are released, they're going to cause type 1 hypersensitive reactions in that individual. Now, what are allergens? Now, as I mentioned before, allergens are a specific type of antigen. Now, say suppose when an antigen enters inside the body, what happens is B cell gets activated, they produce antibodies. The main function of these antibodies either is to neutralize the antigen or it is going to mark these antigens for elimination. Whatever it is, At the end of the reaction, when the pathogen has been removed from the body, the concentration of antibody also decreases. But there is one particular disorder which is called as atopy. And atopy is not found in all the individuals of a given species. Some of the individuals of the species are going to have a condition which is called as atopy. And what is atopy? In such individuals, there's an abnormally high level of circulating IgE as compared to a normal individual. I hope you have followed, which means atopy is what again? It is nothing but that these individuals are going to have abnormally high, abnormally high level of IgE circulating in his bloodstream as compared to a normal individual. But you have to understand that in a normal individual, the amount of IgE is always very less as compared to any other class of antibody. IgE is found in very less concentration in the bloodstream. The person having atopy, what is wrong with them? That they have abnormally high levels of circulating IgE in the bloodstream as compared to a normal individual. And atopy is a hereditary predisposition, all right? So it is a hereditary predisposition to the development of immediate hypersensitive response towards common environmental antigens. And such individuals allow these allergens to induce inappropriate IgE production, leading to tissue damaging type 1 reaction. As I mentioned before, the B cells, when they interact with an allergen, they will specifically produce IgE. Did you understand? So these atopic individuals, what happens in these individuals? The allergen, when enters inside the atopic individuals, they will induce inappropriate IgE production, leading to this type 1 hypersensitive reactions. All right. So you have to remember that these allergens are nothing but these are antigens only, very special type of antigen. And when this allergen enters, what is going to happen? They're going to lead to inappropriate production of IgE only. Did you understand? And there is one condition in this individual which is called as atopy. Now, what is atopy? Atopy is nothing but in these individuals, the IgE level is abnormally higher. Okay, the IgE level is abnormally higher as compared to a normal individual. We have to also understand that atopy is partly genetic, which means it runs in the family. That means someone in your family will also have an allergic reaction. May not be the same type of allergic reaction, may be a different reaction, but it basically runs in the family. So it is basically partly genetic. And these individuals, they're very sensitive towards allergic reactions like hay fever. Now, hay fever is also called as rhinitis, and it is related to upper respiratory tract. Like many people, they complain that they have dust allergies. Then some people complain that they are allergic to your pet hairs. So these are all basically called as hay fever or rhinitis. So it is related to your upper respiratory tract. Then we have asthma. As we all know, what is asthma? Asthma is nothing but it is a breathing problem. Again, it is related to our respiratory system, but this time it is not upper respiratory tract. It is basically a lower respiratory tract that includes our lungs. Did you understand? So lungs is the place where our gases exchange like oxygen and carbon dioxide. The breathing, we breathe, it goes inside the lungs. So asthma is nothing but it is a breathing problem. So obviously it is related to our lower respiratory tract that includes the lung. Then we have eczema. Eczema is a dermatitis. It is a skin-related disorder which is commonly seen in kids. We also have to understand that most of these allergic reactions, they usually occur in the mucous membrane surface of our body. So wherever we have this mucous lining, the allergic reaction occurs in those parts of our body. And these allergens, either we may have ingested them, like say suppose as I mentioned, many people they have food allergies. All right, so either we have ingested these allergens or maybe we have inhaled these allergens through our nostrils. So either way, when it enters inside the body, they're going to show a lot of allergic reactions in our mucous membrane surface of our body. I have given you some common allergens, a list of common allergens, like we have proteins. Many of these proteins are going to act as an allergen, like foreign serum, then vaccines, all right. Plant pollens. Now, plant pollens, they play a very important role during a dry season when the plant starts releasing all these pollen grains. So I have given you few examples here. Drugs, many people are allergic towards many antibiotics. Penicillin is one of them. Local anesthetics, etc. So when I say people are allergic towards drugs like penicillin, like, if anyone has to go for a major operation, the doctor is basically going to check on his skin. The person is allergic to any kind of drugs that they're going to administer. So that is, they try to find out if the person is allergic or not. Many people are allergic to foods, especially nuts and seafoods, eggs and beans. It can be any form of food, okay? Excuse me. Insect products like bee venom, when bee stings, wasp venom and venom, they can also be allergic. Moles pose, like as I mentioned before, animal hair and danders. Many people are hyperallergic to your pet hairs. So they start sneezing, coughing, runny nose, water comes out from their eyes. As I said, it is all called as hay fever. Now, what are the cells that is involved in type 1 hypersensitive reactions? Now, as I mentioned before, you have seen in a diagram also, there are two types of cells which is playing an important role in causing allergic reactions. These are mast cells and basophils. Mast cells and basophils, they have a granular cytoplasm. That means they have granules in the cytoplasm that carries a pharmacologically active mediators. So when the degranulation of the membrane of these cells occurs, these active mediators release from the granules into the surrounding medium. And these pharmacologically active mediators are going to have the type 1 tissue damaging type 1 hypersensitive reactions. Now, basophils and mast cells, these are blood cells only, which are obviously like other blood cells are produced inside the bone marrow. Basophils, once they are produced inside the bone marrow, they are released into the bloodstream where they're going to circulate. The mast cells are also produced in the bone marrow, but they release into the bloodstream in an undifferentiated form. They migrate towards peripheral tissues such as connective tissue, skin, mucous membranes of gastrointestinal and genitourinary tract, where they undergo differentiation and they form mast cells. All right, so you have to remember this, that basophils, they're produced inside the bone marrow, they release into the bloodstream, circulate in the bloodstream. Mast cells are produced in the bone marrow just like a basophils, but they release in an undifferentiated form. But once they migrate towards your peripheral tissues, they differentiate into mast cells. Now we have come across a point where we have discussed that the IgE is going to bind to their specific receptors which are expressed on the surface of basophils and mast cells. So we have to understand that these cells, that is basophils and mast cells, are going to have two types of receptors for your IgE. One is called as the high affinity receptor, which is designated as FCER1, and a low affinity receptor, which is called as the FCER2. What is the difference? Difference is very simple. High affinity receptor means the antibody is going to bind to its receptor tightly. Low affinity receptor means the antibody is going to bind to its receptor weakly. And for high affinity receptor, even a very small concentration of IgE is enough. Whereas for your low affinity receptor, you need more concentration of IgE so that it binds to the receptors present on the surface of basophils and mast cells. Now, this is a very important event during an allergic reaction that is called as degranulation of the membrane. Let us discuss what basically happens. I've been telling you that degranulation of the membrane of both your basophils and mast cells will release huge amount of pharmacologically active mediators. Now, why does degranulation of membrane occurs? These degranulation of membrane occurs due to cross-linking. All right, so you have to understand that degranulation of the membrane of basophils and mast cells occurs because of cross-linking of IgA, which is already bound to their receptors on the surface of basophils and mast cells. Now, we've been hearing about allergens, which is going to cause cross-linking. But it is not only allergen that will cause cross-linking. There are certain other compounds which can cause cross-linking, like we have various drugs which can cause cross-linking of the IgE bound to the surface receptors. There are certain anaphylatoxins also, which can cause cross-linking of IgE on the surface of basophils and mast cells. Anaphylatoxins that include C3A, C4A, C5A, and various drugs. Did you understand? So the main important thing is that degranulation of the membrane of basophils and mast cells. And how does degranulation occurs? It occurs because of cross-linking of IgE, which is bound to the specific receptor present on the surface of basophils and mast cells. And you have to also understand that it is not only allergens. It can be various drugs, it can be anaphylatoxins. But what is the main thing we have to understand? That it is a cross-linking of IgE, which is found on the surface of these cells. So what happens right after the cross-linking? Right after cross-linking, the membrane of these cells are going to open up channels for your calcium ion. Now, why this calcium ion is very important? First of all, we have to understand that the concentration of calcium ion is very, very low inside the cell. Usually, the calcium ions are found outside the cell, or in case of muscle cells, they are found in the sarcoplasmic reticulum. And what is the function of calcium ions in sarcoplasmic reticulum or muscle cells? These calcium ions are going to help in muscle contraction and relaxation. But usually a normal cell will not have calcium ions inside the cell. So here what happens? Immediately after the cross-linking of IgE, what is going to happen? The cell is going to open up ion channels so that calcium ions can enter from outside towards the cytoplasm of the cell. Once the calcium ion enters inside the cell, the concentration of calcium ion automatically increases. All right, so calcium ion is entering inside the cell, the concentration increases. This high concentration of calcium ion is going to lead to the production of a very important fatty acid, which is called as arachidonic acid. Now, what is the importance of arachidonic acid? Now, arachidonic acid is going to help in the production of two classes of very effective mediators. These are called as prostaglandins and leukotrienes. All right. The increase in the concentration of calcium ion is not only going to help in the production of arachidonic acid, it is also going to help in the movement of the granules. Remember, I told you basophils and mast cells, they have a granular cytoplasm. So increase in the concentration of calcium ion is also going to help in the movement of granules, and it also helps in the fusion of these granules to the membrane of the basophils and mast cells. So once it fuses to the membranes, what is going to happen? We all know degranulation occurs, and the active mediators are released into the surrounding medium. Did you understand? So you can see the calcium ion plays a very important role. Now, in order for the calcium ion to enter inside the cell, what we have to do? First of all, the cross-linking of IgE sitting on the surface of basophils and mast cells are important. And who is going to cause cross-linking? It can be allergen, as I mentioned, it can be anaphylatoxins, it can be various drugs. All right. So they're going to cause your, cross-linking. As soon as cross-linking occurs, within seconds, what happens? Ion channels for calcium ion opens up. Calcium ion enters inside the cell. The concentration of calcium ion increases, which helps in the production or formation of arachidonic acid. What is the importance of arachidonic acid? It helps in the production of your prostaglandins and leukotrienes. Increase in the concentration of calcium ion also helps in the movement of granules towards the membrane of the basophils and mast cells. So once it fuses with the membrane, then what happens? We all know degranulation occurs and release of various pharmacologically active mediators in the surrounding medium takes place. So this is the whole process, pictorial representation. So we have the antigen, they have written antigen, it is actually an allergen that leads to cross-linking of Ig, which is bound to the specific receptors, then calcium ion enters inside the cell, that leads to the movement of these granules, it fuses with the membrane of the cell and degranulation, okay? So these mediators are released, these mediators are going to cause the tissue damaging type 1 hypersensitive reactions. So this is a pictorial representation. Now what are the different mediators? Now these different mediators, when are released by the basophils and mast cells, they usually act on local tissues and cells. All right, they act on the surrounding, tissues and cells such as eosinophils, neutrophils, T cells, monocytes, and platelets. All right, so they usually act on the surrounding medium. Now there are two types of mediators. One is called as the primary mediators, another one is called as the secondary mediators. Now what is the difference? Difference is very simple. The primary mediators are those which are produced and stored inside the granules. Did you understand? So here, even say suppose a mast cell and basophils, they are inactive, they are not active, all right, they're inactive. In an inactive basophils and mast cells, these primary mediators will be produced and they will be stored inside the granules. Did you understand? And once these cells undergo activation and degranulation, then these mediators are released into the surrounding medium. So what is secondary mediator then? Secondary mediators are those which are produced only after activation of the mast cells and basophils or after degranulation. All right, so that is the basic difference. Primary mediators, they are produced and stored in the granules before degranulation. Secondary mediators are those which are produced after activation of the mast cells or basophils in response to an allergen, or maybe after degranulation of the basophils and mast cells. All right, so they are produced later on. I have mentioned examples here like histamine. So now you know, we have been hearing about histamine a lot of time. Now we know histamine is a primary mediator. Apart from histamine, we have proteases, different kinds. We have eosinophil chemotactic factor, we have neutrophil chemotactic factor. So these are the examples of the primary mediators. These are produced and stored inside a granule. What are the secondary mediators? These are produced after target cell activation and degranulation. Examples I have given you: prostaglandins. We have come across prostaglandins before. We have come across leukotrienes. And apart from these two, we have bradykinins and various cytokines. So these are the examples of secondary mediators. Now, once the histamine is released, how can we see the biological effects of histamine? That the biological effects of any molecule is seen when it binds to its own specific receptors. Histamine is no exception. So histamine is also going to act once it binds to its own specific receptors on the surface of the target cell. And these receptors has been classified as H1, H2, and H3. So we have to remember that it is not only histamine, any kind of molecule to see its biological effects, they have to bind to the specific receptors which are expressed on the target cell. So histamine will also have its biological effects when it binds to the respective receptors which are present on the surface of the target cell. And this has been categorized as H1, H2, and H3. Now, what are the effects of the histamine? They will cause smooth muscle contraction. They will increase the vascular permeability, which means the permeability of the membrane of the blood vessels will increase so that cells can migrate from the uh, from the bloodstream towards the affected tissue. Increased mucus secretion. So these are the effects of histamine. Now, the biological effects of prostaglandins and leukotrienes is also seen when they bind to their own respective, receptors. But their effects are very effective. All right. So you have to remember their effects are very effective and they are long-lasting as compared to your primary mediators. So primary mediators will show immediate effect. The secondary mediators, which includes prostaglandins and leukotrienes, they're very effective and their effects are long-lasting. But you have to remember that their biological effects can be seen only when they bind to the respective receptors. Now, what are its effects? Like leukotrienes are going to mediate bronchoconstriction. What is the meaning of bronchoconstriction? That means it is the constriction of the bronchioles, and that is going to, block your airway. So we have breathing problem. Increased vascular permeability that we have discussed already. That means increasing the permeability of the blood vessels, so that the cells can move from the bloodstream towards the affected tissue, and mucus secretions. What about prostaglandins? Prostaglandin also is going to mediate bronchoconstriction. So this is all about your type 1 hypersensitive reaction, the first part. In the second part, we will discuss about the types of type 1 hypersensitive reaction. That means we're going to discuss about examples. Thank you.